Okay, so my task now is to introduce you to the mycotoxin issue. Many of you are aware of that issue anyway, otherwise you wouldn't be here, I guess. But this is also kind of as a starter, so to speak, for the Mycotoxin Summer Academy, and where I would like to highlight a couple of actual cases, also mycotoxins in the context of risk assessment will be addressed. I will also be dealing with the history and effects of mycotoxins. However, that will be dwelt on in the lecture given by Mark Lemons after the coffee break in more detail. I will also briefly introduce you into the chemistry of mycotoxins with a couple of occurrence data, also briefly touching the regulatory side, and finally we'll round off this presentation with pointing out the need for prevention and detoxification. Well, the actuality of the mycotoxin topic has clearly been visible through a couple of very recent cases with reports on contaminated Serbian corn supplied to German farmers. In fact, actually, Alfred Töpfer International imported 45,000 metric tons of corn contaminated with aflatoxins from Serbia. A batch had been delivered to more than 3,000 farms northwest of Germany for animal feed, which has actually even led to reports in The Guardian pointing out that the Germans test milk in carcinogen scare as a result of aflatoxin B1 contamination. Possible contaminated milk products contaminated with aflatoxin M1, the metabolite of aflatoxin B1, was likely to occur, which was actually not the case in Germany, but in Serbia. And there has also been reports which you can actually look up in YouTube as well, with these contaminated corn, which was imported to Germany from Serbia. So no need to emphasize anymore that actually this is a topic which has not been there for many, many years, but has actually still got its actuality. So with the mycotoxin issue, we're actually right in the middle of the topic of food and feed safety, and that is why I would like to, in the beginning, to define, so what is actually food safety? And this is actually about ensuring that food is free from microbiological or chemical contaminants that might cause harm to human health. It's kind of obvious. Feed safety is about ensuring that feed is free from microbiological and chemical contaminants that might cause harm to animal health. But in addition actually to the definition of food safety, here we have got this additional aspect, or transfer residues to animal products, milk, meat, etc. So by alluding back to the case of aflatoxin B1 in the maize imported into Germany from Serbia, which was contaminated with aflatoxin B1, a transfer residue to animal products would have been aflatoxin M1, which you find in milk and possibly also in meat products. So a feed safety issue could easily also transfer into a food safety issue. But there are actually a couple of chemicals that may enter the food supply. These are, for instance, food additives like preservatives, sweeteners, etc. There are also other food contact chemicals such as food packaging materials, as for instance BPA, bisphenol A, which can migrate actually from the polycarbonate plastics into the water, processing aids and so forth. There are also a couple of agrochemicals such as pesticides and veterinary drugs which may enter the food supply. And most importantly for the Mycotoxin Summer Academy, the natural toxins, fungal, algal, microbial plant toxins, which we'll certainly elaborate in any detail here in our academy. There's also adulteration, where actually a contaminant has been added for the purpose of pure greed, to maximize the profits to fake nitrogen and protein content in milk powder by the addition of nitrogen-rich melamine. Coming actually from pre-market to post-market, there are also processing-induced chemicals like acrylamide, fluorine, chloropropanols, also hidden allergens like peanuts in chocolate, which has not been declared as peanut-containing chocolate. And last but not least, environmental contaminants such as metals, POPs, so-called persistent organic perfluorinated or perchlorinated compounds. So this is actually the background of chemicals which may enter the food supply. There has been quite a lot of scientific development in food safety, and especially in analytical chemistry. I am able to tell you that the techniques and analytical methods which are there have tremendously advanced in pace. We can actually detect smaller and smaller amounts of chemicals in the foods, down to the ppb, to the ppt, parts per billion, parts per trillion range. However, in contrast to analytical chemistry, where we're able to detect lower and lower amounts of chemicals in the food and feed supply, toxicology is advancing actually only very slowly. While this is also clearly visible through the difficulty in getting approval for animal experiments on the one hand, but also by the great effort and the great undertaking in order to specifically characterize mycotoxins, for instance, for its toxicological effects. So the consequence is actually we know lots about what is present in our food, but little about actually the toxicological significance. And this is actually not going to change in the next couple of years. However, there is now high throughput screening available. This is all done on microtiter plates. There are alternatives to animal experiments on the way. But still, we can summarize that risk assessment is still in its infancy. And that is a kind of a picture which shall outline biochemical hazards and a general scheme for risk assessment of such biochemical threats. It actually all starts with hazard identification, which can be something like a shark, where you clearly identify this as a potential hazard, but you have no actually clear vision, assuming that you have never seen a report about the health-threatening effects of a white shark. And the white shark could also be a pesticide, or could be a new mycotoxin on your radar, which has been identified with your LCM SMS, where you can only say at this stage, well, there is a potential new mycotoxin there, a secondary metabolite, a hazard has been identified, but has in the next step to be characterized. Well, in the case of shark, it can obviously bite you, it can harm you. In the case of mycotoxins, you would actually also have to characterize this hazard with proper toxicological effects, studying the toxicological effects of these mycotoxins, and as such then moving on from the identification to the characterization of that hazard. And finally, it's about exposure assessment. So whereas the shark has clearly been identified as a hazard, the probability of an encounter is actually very low. The question is kind of what is the probability of encountering or being exposed to mycotoxins? That is something which has to be sorted out by either biomarker studies, which you will also be witnessing in the next 2 weeks, by checking actually for biomarkers, mycotoxins, mycotoxin metabolites in the urine, or by actually checking the contamination levels of food and feed items in conjunction actually with a consumption behavior. So how much of the food, how much of the feed is being consumed, what is the contamination level, and then actually you can perform an exposure assessment, feeding into risk characterization, and finally ending with proper risk management, where actually it could be a labeling, a shark has been sighted, or it could be a labeling of the food that an allergen is there, or regulatory limits and the implementation of these limits. Well, how does the situation look like in Europe? We've got the European Food Safety Authority, which deals with risk assessment, including mycotoxins. Large safety factors are actually used to protect human health by a factor of 10, for instance, for species differences, another factor of 10 for individual differences, etc., etc. This will be elaborated in Mark Lemon's presentation. What I would like to point out at this stage, that the precautionary principle is used where no adequate toxicology is available. So in case of the doubt, you assume the worst. And that is actually done by EFSA scientific panels. There is one dealing with animal health and welfare. There are panels dealing with food contact materials, and most importantly for the Mycotoxin Summer Academy, the so-called Contam panel, which deals with contaminants in the food chain. And each panel consists of various working groups, and there is actually currently a working group on Fusarium toxins, which I am actually part of. So specialists from all over Europe meet, let's say, four times a year, have an additional 4 to 6 times, depending on the necessity, conference calls where they discuss individual opinions on individual toxins. Currently, actually, an opinion on nivalenol has been issued just last week, and the contaminant working group on Fusarium is now working on an opinion dealing with enietins and bovarecin. Well, what I would also like to point out at this stage, that we have to distinguish between residues and contaminants. So please note that actually a man-made chemical contaminant can be considered as a residue. We are adding a pesticide on purpose. We are adding a fungicide on purpose to prevent fungal growth. And then actually what's left there in the food after washing, after cleaning, after processing of your food item is a residue. So that could also be a veterinary drug. Whereas a contaminant is, for instance, a natural toxin. So this is actually this has not been added on purpose. This is actually a contamination during food production. Could also be during food packaging contamination, or is there because it cannot be prevented, like for instance a Fusarium toxin. So that is also something which is often mixed up. So biochemical hazards have also been considered in view of climate change, in view of global warming. Let me point out actually that the natural toxins rates of production can strongly be influenced by environmental as well as climate changes. So air pollution and rising temperatures are causing different profiles and also occurrence for natural occurring algal blooms in the ocean and freshwater. So there have been reports, for instance, in Alaska, in Canada, with the death of several Canadians as a result of unexpected occurrence of domoic acid in shellfish as a result of unexpected algal blooms, which have been very intense and have never been encountered or reported before. And there are actually generations of Canadians who have consumed certain shellfish somewhere in Alaska. and all of a sudden they are being asked to be aware of domoic acid. Often people don't believe that because they say, well, we have been consuming these shellfish for many generations, but before global warming, obviously global warming is also strongly influencing the phycotoxins, as for instance domoic acids, which are finally found in shellfish and can be poisonous to humans. Well, with mycotoxins, we are certainly in the area, right in the middle of the area of biotoxins, or you may also call them natural toxins. Within the natural toxins, we've got phycotoxins, which I've alluded to before, which are produced by algae, which can be found in shellfish. We've got plant toxins, alkaloids, or also pyridostigmine alkaloids, among those toxins produced by plants, which are often not as toxic as phycotoxins or mycotoxins, but are still actually species dependent. For instance, pyridostigmine alkaloids can be of severe toxic importance for horses because they are liver toxins. Then we've got bacterial toxins, which are of considerable interest because they show the greatest toxicity of all these toxins mentioned here. Besides maybe zootoxins, however, those are only relevant for those of you who are keeping poisonous snakes at home. Is there anybody among the audience who is keeping poisonous snakes? Well then, if not, then you will have to worry more about the other categories here, and one of these categories are certainly mycotoxins. So what are mycotoxins? And there is this guy from Ice Age, and he says maybe aflatoxins in nuts. And it's kind of true because aflatoxins have been used kind of as a synonym for mycotoxins because they were the first mycotoxins which were discovered in the 60s. Now we have a clear definition of what mycotoxins are, with deoxynivalenol, by the way, is one of the major representatives here in Europe. These are toxic secondary fungal metabolites. So they are toxic and they are actually a result of the secondary metabolism. This is usually the results of some stress these microorganisms are exposed to. For instance, they don't have sufficient nutrients available. Now they define a secondary strategy by excreting toxic compounds which can weaken the immune system of their host, the plant. The plant is weakened, cannot actually resist the fungal invasion, and the fungus can grow more easily and rapidly as a result of the excretion of these secondary metabolites, mycotoxins, and can further take advantage of the nutrients provided by the plant. So Aspergillus species are among the most important ones found all over the world, particularly in the hotter regions of the world, especially humid and hot regions. Penicillium species, all these species will be alluded to during the coming week. What I would like to point out here is the prevalence of Fusarium species, which are the producers for trichothecins, and also deoxynivalenol, which are the most dominant species in the moderate climatic regions of the world. A few words on history. So the epidemics of ergotism has been already reported 857 Anno Domini, actually resulting from ingestion of cereals infected with ergots, with actually the so-called holy fire as a result of the burning sensations as a result of reduced blood flow, which actually can even end up with people having lost their limbs as a result of no proper blood circulation. There have also been lots of reports from medieval history on poisoned domestic animals after ingestion of feed contaminated with fungi. But the whole story actually started in the 60s with the so-called Turkey X disease. X standing for, well, we don't know the reason for this particular disease. Actually, 300,000 turkey died as a result of consumption of peanut powder infected with Aspergillus flavus, as they found out later, and as a result, actually, the highly toxic fungal metabolite aflatoxin was isolated, and intensive research on other fungi and toxins produced by those species and also other species took place. Well, this is actually quite well known. In meanwhile, what is not so well known actually is that the first report of emesis and other symptoms caused by Fusarium infected grain has been reported in the Tierärztliche Wochenschrift, which is actually a weekly magazine of vets by Miesner, and actually David Miller from Carleton University. He sent me this old journal or this report and asked me to translate that. And as a result of that translation, we found out actually that this is in fact the first report where actually farmers from northern Germany complained about feed refusal of swine plus, under quotation marks, some signs of sickness when feeding those with poisonous US barley. So although actually the animals seemed to like the feed in the first instance, they refused subsequent feed rations. And actually, already back then, Fusarium roseum, which is the sexual form of Fusarium graminearum, was identified as the poisonous agent of the poisonous barley, and Fusarium causes, and that actually, well, that was the explanation, Fusarium causes degradation of the infected commodity, which forms substances that may interfere with the digestive system. So far the state of the art in 1929. And actually even more interestingly, as a result of that finding, there was another report by Dankwart in 1929 on chemical analysis of this poisonous barley. And they have investigated what is the toxic principle in the poisonous US barley. And they found out actually here that the intoxication might have been caused by volatile compounds of hydrocyanic acid or plant toxins such as fungi. So there was an assumption which was not, which they were not able to confirm. Actually, quite interestingly, hydrogen cyanide was used as a pesticide at that time, and there were actually public concerns that residues of this hydrogenic acid may remain in the food, in the grain, but they were actually not able to determine hydrocyanic acid anyway. Then they came up with the idea maybe there are biogenic amines as a result of protein degradation caused by fungal infection, or alkaloids which cause actually the observed toxicity. So however, neither of these substances were detected in this US barley, and they also observed actually that the US barley shows a different fluorescence compared to German barley. So from time to time, green light spots were observed under UV light. So the conclusion back then was there is no method available that can differentiate between good and bad barley. So I would say, poor guys, there was no LCMS available at that time. But actually, it's quite interesting, actually, 80 years ago, what the state of the art was back then. And this is actually kind of an historic document, because these are really the first reports in Europe which deal with Fusarium. So what is the nature of these mycotoxins? And you see actually major representatives of these toxic secondary metabolites. They show different polarities, from nivalenol being very polar to serralenone, the estrogenic mycotoxin, which is actually apolar. We have different UV absorption fluorescence, where although we are able to say that any substance which shows fluorescence shows also UV absorption. Not any substance which shows a UV absorption shows also fluorescence. So zearalenone is a fluorescence molecule which can also be detected by UV absorption. Ochratoxin A is also showing fluorescence, whereas, for instance, deoxynivalenol does not show any fluorescence at all. However, it is a UV absorber, with actually T2 toxin as a major A-trichothecene being a non-UV absorber. So actually, that is a tricky when you have got an HPLC UV methodology to also cover T2 toxin because it's such a weak UV absorber. So certainly you can work with post-column derivatization also in order to enhance the UV absorbance. They also differ in their ionic nature depending on the pH. So for instance, for moniliformine, that actually can occur as an ion. We will also see for monosins that this is the case. They occur in different commodities, whereas aflatoxin can easily occur in peanuts, in pistachios. Deoxynivalenol is mainly found in grains. And we've got actually different concentration levels of interest according to the different toxicological effects of these mycotoxins. Aflatoxins are already of great importance in the lower microgram per kilogram range, whereas deoxynivalenol is a toxin for which regulations exist in the ppm range. That shows you a further picture on the, with additional representatives, including for monosin. You see actually these carboxylic functions, and you can actually easily imagine that actually the ionic nature also depends on the pH. Actually, at lower pH, you would actually reduce the dissociation of the carboxylic function, and as such, you will actually observe ionic nature at higher pHs and not at lower pHs. Bovaericin, actually a cyclic hexadeptipeptide, which with carbonylic with CO functions pointing towards the center of the molecule, which can actually staple up and can transport alkali ions, for instance, through these ion channels, through membranes, and can even be cardiotoxic, and that is still under investigation. So these are apolar, polar acidic, and also polar or basic nature. You can actually see here nitrogens, secondary amino functions which actually show basic nature. So you can actually already imagine that a routine method which covers all these different mycotoxins with one extraction, one cleanup, and one detection strategy is almost impossible, and that is why different strategies and or a very sophisticated methodology like a tandem mass spectrometry has to be applied. Well, this does also show you the mycotoxin pattern of here in this case for Canadian durum samples. When you look actually in the first at the first couple of toxins, deoxynivalenol, nivalenol, zearalenone, HD2 and T2 toxin. So this is kind of, that's the common picture here. So you can actually see here, so that is about 100 microgram per kilogram. This is 1,000 microgram per kilogram. That actually you see here the common fusarium mycotoxins. However, when you increase the screen by also covering anietins, moniliformine, and so forth, you see that there is stone glucoside there are anietins, there is moniliformine there actually. So the picture of potential mycotoxins, secondary metabolites, which can potentially occur in samples is much bigger than to those toxins which are regulated. Even alternaria metabolites were found in these Canadian durum samples, and also other less prevalent toxins like ergot alkaloids. So 57 different fungal metabolites have been identified in these Canadian durine samples, and there are reports which even report about a greater number of these toxic metabolites. And to make the story actually even more complicated, mycotoxins can be metabolized, and you will also hear during this week about metabolization of deoxynivalenols, the most important process taking place in plants, with deoxynivalenol as the major representative, with its epoxide ring, which is actually the reason for the inhibition of the protein synthesis and the weakening of the immune system, which is clearly a toxic molecule and can, through the help of UDP glucose and glucosyl transferases, an enzyme which can be found in almost any plant, be conjugated to DON3 glucoside. So you've got the glucose here, which is the so-called non-toxic or masked deoxynivalenol, masked because this usually escapes routine analysis because of these polar compounds. You will usually lose that substance with any cleanup because it will remain there on a polar column and will not be eluting. So this so-called detoxification process takes place through glucosylation and is actually an important aspect also when considering Fusarium head blight vis-a-vis healthy plants. So it seems that those wheat and maize genotypes which have got a genetically based resistance against Fusarium infection, against mycotoxins, have a greater ability to express glucosyl transferase and to perform that detoxification process in plantar. Actually, obviously, to make this molecule more polar and also to be able to rinse it out or to make it better soluble in water and to more quickly and more easily get rid of these toxic substances. Well, we have now been able at the IFA Tulln to trace that the ability of this glucosyl transferase back to a certain QTL, a so-called quantitative trait locus on a wheat chromosome. So actually we are now able to pinpoint this genetic region of a chromosome, which is responsible for glucosyl transferases, and that actually helps us certainly also to breed for resistance genotypes by selecting these wheats which have got that particular QTLs. And what is actually most important for food safety is the fact that DON3 glucoside can potentially, through hydrolysis but through enzymatic cleavage, again get rid of the glucose and release the parent toxin and cause intoxications where you actually would have not expected these intoxications to occur. Let's say you've got food which has a certain level of deoxynivalenol but with an additional not known level of DON3 glucoside, then actually you would consume this food or feed when you give it to animals in good faith that there is actually not a high level of toxin there, and then actually these not detected mast toxins are cleaved, are hydrolyzed, are releasing the parent deoxynivalenol, and then you have got some inexplicable cases of intoxications because you were not aware of this additional amount of deoxynivalenol. And that is what we summarize nowadays as so-called emerging mycotoxins. I personally prefer the expression neglected mycotoxins because ergot alkaloids or N-etins have been around for many decades. However, my take actually on emerging toxins is that we have got actually new unexpected occurrence data, as a result of climate change. A good example is domoic acid. A good example is also the occurrence of aflatoxins in Serbia imported to Germany, that actually all of a sudden you find aflatoxins in moderate climatic regions where actually hardly any aflatoxin has been reported before. And a very important component within emerging mycotoxins are also advanced analytical methods, which can also lead to new unexpected occurrence data, but which can also lead in that direction to new findings, such as, for instance, the presence of masked or conjugated mycotoxins. And to define these emerging mycotoxins, there is proliferin, bovaricin, enietins, moniliformin, and any potential other mycotoxins, which I will also be dealing with in the afternoon during the state-of-the-art session. And we can, from our own research, confirm that enietins, moniliformin, alternarium metabolites do frequently occur in countries with moderate climate. However, several other mycotoxins are prevalent as well. For instance, especially from samples from tropical countries, phosphoric acid, stigmatocystin. There's actually a new occurrence survey on the way, which EFSA has called for, amodin, and several others as well. The question actually is, what is the impact of these compounds? What is the impact of these emerging toxins which are not regulated so far? There is also the question of additive or synergistic effects. For instance, what we do know is that the co-presence of non-toxic phosphoric acid increases the toxicity of deoxynivalenol. So there are actually lots of questions there which still have to be answered, particularly in view of this emerging field. So molds and toxins, would also like to summarize here factors influencing the fungal growth and toxin production. So clearly weather conditions like temperature, humidity are of crucial importance, particularly in the area of Fusarium. In order to fight Fusarium, there's actually also the possibility of adding fungicides, which have only have only some minor effects. However, if they are applied at the right time, just a few days after blossom, then actually they can have a much more pronounced effects on the reduction of Fusarium growth. That is why the applications of fungicides is usually sold by companies like Bayer in combination with so-called weather stations, which monitor humidity, which monitor the temperature to really pinpoint. So when is the exact time and date? What is the correct timing for applying those pesticides? Tricky, not always successful, and there's also a variety of grain which does certainly also affect fungal growth. Insect manifestation, the most important one being the corn moth. The corn moth actually invades the corn, bores a hole, drills a hole into the corn cob, and as a result of this hole, the fungus actually can much easier penetrate and grow into the corn cob, and as a result, a combination of the presence of the corn moth and Fusarium or any other fungi is resulting in much severe effects. Also, as far as mycotoxins are concerned, crop density. So we are actually now dealing with extremely high crop densities to get the most out of our agricultural activities. Fertilization also an effect. When we are dealing with harvesting, crop maturity, moisture content, well, clearly you would actually shall harvest at a time point where the moisture content of the grain is as low as possible. Well, that's easy to tell an Italian farmer. It's actually much more complicated, the situation, when you tell the same thing to a farmer from Scandinavia, where it's almost impossible to reach a moisture content which is below this magic 13%, which is the moisture content at which you can expect that no further fusarium growth takes place. Certainly any agricultural practice does influence the fungal growth and toxin production. Again, here, insect control for the storage, moisture preservation. So we have to distinguish between those fungi which grow particularly on the field, like fusarium and its toxins, and those which are more a storage problem, including penicillium, which produce, for instance, ochratoxin A. So proper storage is a very important aspect. And finally, the distribution. What are the shipping conditions, processing? So these are all factors to be considered, and that could be a lecture on its own, but this is just an introduction to the mycotoxin issue, and where I would also like to summarize some effects of commodities once they are invaded with the fungi. You've got here typical signs of Fusarium head blight disease. Here you've got a wheat ear with its kernels. So typical bleaching effects, and this is also makes quite obvious when you look at these bleached Fusarium head blight affected kernels versus these healthy kernels, that you can actually easily imagine that these kernels are not of the same quality as as healthy kernels. They actually lack the same high amount of protein, so they are lacking protein quality as well. Baking quality is reduced, and you can actually imagine that besides this food and feed safety issue, this is also an economic issue, resulting in this magic number of this $932 million a year, almost $1 billion of US dollars lost as a result of mycotoxin contamination, which has actually by the Food and Agricultural Organizations been defined as, well, 25% of all the agricultural commodities are contaminated with mycotoxins. But this is actually a finding which dates back to 25 years ago. I would say with LC-MS technology, we can now say that at least 90% of the commodities are contaminated. It's just a question kind of which contamination levels are we talking about here. And we've got also other affected commodities like infected maize. You've got here a corn cob with severe Fusarium infections. The good thing for us chemists, and that is why we have also a second week dealing with LC-MS and analysis of mycotoxins, is the fact that a corn breeder, a corn grower is not able to distinguish between the contamination level of mycotoxins of this particular corn vis-a-vis this particular corn cob. This actually, the actual contamination level of mycotoxins has to be verified with chemical analysis. This cannot actually be done by just visible inspection of these corn cobs. Why? Because different species, different strain of different species, like Fusarium graminarum, there are different strains around, have different abilities of or capability to produce mycotoxins, and that is why chemical analysis is of utmost importance, certainly also for these more severe cases, which are also depicted on these lower pictures. Last but not least, another affected commodity, which is more important in the area of aflatoxin contamination with Aspergillus, is peanut, and is certainly of great importance not only for feed but also for food industry. Well, how do these affected commodities translate into toxicological effects? This is kind of just an intro slide for the lecture to come by Mark Lemons. We've actually got retarded growth of chicken, for instance. We've got pale liver syndrome as a result of aflatoxin contamination. We've got here swelling of vulva of swine and atrophy of sperms as a result of the effects of estrogenic mycotoxins, as for instance zearalenone. However, all these pictures date back to something like 30 years ago, where mycotoxin research was focusing mainly on heavy effects. So kind of, okay, so I want to know what is the effect of deoxynivalenol, of zearalenone, of aflatoxins, and then I treat the animal with the highest dose possible in order to see some nice effects. I don't want to say that you will not be able to see the swelling of vulva, for instance, in very exposed areas, for instance, in developing countries. But nowadays, research is dealing with background exposure. So background exposure to mycotoxins, especially as food safety, but also what feed safety is concerned, has become of greater importance. We have actually a lack of information. What is actually the effect of a constant exposure to low levels of mycotoxins, of a cocktail of different, possibly also synergistically acting mycotoxins, in combination even with other chemical contaminants such as pesticides. What is the immunotoxicological effects of constant background level exposure of low levels of mycotoxins? So that is actually the research topic which we are dealing with nowadays. And actually that leads us to mycotoxins or fungicides. I've already pointed out that there is a possibility also of treating the Fusarium issue with the use of fungicides. What is actually important to point out at this stage is that this is just one strategy. It is also a very actual strategy because of all the bee problem of death of millions of bees worldwide. The issue of pesticides has been discussed in all mass media. But it's actually quite interesting when you compare when you look at the toxicity of some fungicides like dupoconazole, which are used to treat Fusarium diseases on the field. They've got an LD50 value of 5,000 milligram per kilogram body weight, and the maximum residue limit in cereals, which is actually allowed, is 50 microgram per kilogram. So how does that actually compare with mycotoxins? You know, deoxynivalenol is not among the most toxic mycotoxins, but it has got an LD50 of 50 milligram per kilogram body weight. And we allow here regulatory levels of 1,250 for cereals. So meaning that deoxynivalenol has actually got an acute toxicity which is a factor of 100 higher compared to fungicide depoconazole. But we allow maximum limits in our cereals which are a factor of more than 20 higher than the levels which we allow for depoconazole. So that makes actually from a scientific, from a logical point of view, not really much sense. Clearly, you can avoid any addition of fungicides, and you cannot avoid the addition of mycotoxins, or you cannot avoid the presence of mycotoxins necessarily. I mean, that's easier, for instance, for ochratoxins than for other Fusarium, than for Fusarium mycotoxins. And certainly, the ideal strategy, which also the European Commission is now much more in favor of, is not only that our food becomes greener, but that can ideally be achieved by cereals with high resistant levels against fungi. So ideally, you breed wheat, maize, and other genotypes, agriculturally important commodities, which have got a high resistant level against fungi, and as a result, a low mycotoxin content. Well, what can we do about this? There's actually the Swiss food regulation of the 13th century. Some of you, I think, know about this, but I still want to mention it. Fish which cannot be sold can be declared as such and is allowed to be sold to foreigners only. So that is certainly a very strange way or medieval way of approaching that problem. Nowadays, we've actually got different strategies. There is in place a European Commission-funded project which is petering out towards the end of the year, the so-called Micro-Red Initiative, Novel Integrated Strategies for Worldwide Mycotoxin Reduction along the whole food and feed chains. There are 25 partners collaborating. Among those partners, there is also us. We were also work package leaders in the area of analytics. 17 countries were represented with a budget of more than 7 million euro. So that is certainly a way of bringing the knowledge together, working together on reducing this issue along the whole food and feed chain, which finally can also contribute to meeting the regulations, but also possibly implementing proper regulations. That can also be a lecture on its own. Let me just summarize here for Europe that 39 nations do have regulations which are known. The European Union has also harmonized limits for a couple of toxins, including aflatoxins, ochratoxin A, patulin in fruit juice, deoxynivalenol, cearalenone, and fumonisins in different commodities at different stages. That is actually a very complex stuff to be discussed. Processed, unprocessed materials. The EU has also food limits discussed for T2, H2 toxins and ergot alkaloids. For T2, H2 toxins, I don't actually foresee a regulatory limit to be introduced soon based on the recent EFSA opinion. For ergot alkaloids, I think situation is particularly interesting because there is only a guideline there for ergot bodies, but not for ergot for the individual ergot alkaloids. One should not forget that the ergot bodies is just a visible sign of the presence of ergot alkaloids, but it's actually the ergot alkaloids which are the chemical compound which matters, for which we have actually no regulations at all. The EU has also feed limits which exist for aflatoxin B1. There is also a feed guidance value for ochratoxin A and some Rosario toxins, and there is lots of discussion going on within EFSA. The next toxins on the plates are, as I mentioned before, anietins, bovaricin, but also conjugated toxins, moniliformin. So these are actually just to give you an outline or an outlook for the discussions to come. This is just an example because I could show dozens of these slides. This Commission Regulation EC from 2006 that actually is setting maximum levels for certain contaminants in foodstuffs. You can actually see here the limits set for deoxynivalenol, for instance, in the category unprocessed cereals other than durum wheat and maize. So you know durum wheat is the wheat for the spaghetti production, which is actually particularly sensitive towards fusarium infection, and usually if you encounter higher levels in durum wheat, that is set for 1,250 microgram per kilogram of unprocessed cereals. We've also got unprocessed maize, with the exception of unprocessed maize intended to be processed by wet milling. So we've got a level of 1,750 microgram per kilogram here. And when we go further down that route, we see that for pasta, a lower limit has been applied for the final product, which is consumed in very high amounts in the food supply as a food product. And we have got for processed cereal-based foods and baby foods for infants and young children, due to the lower body weight, obviously lower levels have been introduced as regulatory maximum limits. Here, actually, only 200 microgram per kilogram deoxynivalenol is allowed to be present in infant food. Well, I also have to mention at this point that there are different levels or different regulatory limits, not only for different mycotoxins, but also different levels are allowed in different countries of the world. So, for instance, in Canada, United States, they are much more tolerant as far as the presence of deoxynivalenol in cereals is concerned. Another example is actually aflatoxin. We've got here for groundnuts, peanuts, and other oilseeds to be subjected to sorting or other physical treatment before human consumption or use as ingredient foodstuff. We've got for aflatoxin B1 a level of 8 microgram per kilogram and a maximum level of the sum of so-called total aflatoxin content of 15, whereas for almonds we've got a level of 12 and so forth. So that is actually showing you the other ends where you also see the great difference in toxicity of fusarium toxins versus aflatoxins. For those of you who are interested to get more detailed information about regulatory limits which are established for mycotoxins, please just Google this Commission regulation. You get actually all the information from there. So how can this, how can the toxin problem be prevented? Well, use of crops with high fusarium resistance. I've already alluded to that before. Avoid narrow crop rotation consisting of wheat and maize. So, well, if you have got wheat in 1 year and next year you grow maize, actually the remaining wheat on the field is the ideal inoculum for the next year when you grow maize. And then actually through rain and wind, actually the inoculum will easily be spread on the new maize, and which is particularly sensitive to Fusarium graminearum. Use high quality fungicide treated seed and usually not infected seed in order to avoid the Fusarium infection right away. Avoid harvesting of infected maize ears for swine diets. Blowing crop debris into the soil reduces the Fusarium inoculum. There has been kind of a blowing ban in Europe or in certain parts of Austria, where there's been discussions on actually that blowing is actually not healthy for the soil because it kind of compresses the soil. On the other hand, obviously there is also an influence on the presence and growth of Fusarium and the growth of mycotoxins. There's still actually a controversial discussion whether blowing is now beneficial or non-beneficial for Fusarium growth is still under discussion. It's not as easy as I've mentioned before, that saying, well, blowing is good or not good. There are controversial reports on that. Proper storage without delay, well, I think that's a quite obvious thing. There's also decontamination strategy, which I would actually define as kind of, that is a prevention. These are all prevention strategies. This is what I would call end of the pipe technology, right? Because once you have to deal with contaminated crops, how can I get rid of these contaminations at the end? There is physical strategies like washing, segregation, sieving, dehulling. They are either not very effective. Well, with dehulling, that's maybe one of the most effective ones, which is done anyway. Well, with dehulling, you have got the bran, which is healthy but has higher levels of mycotoxins in wheat products, for instance. But mostly, well, when you think of washing, you have to wetten the grains, so this is actually not very practical and also very quite expensive. Equally expensive is ozone treatment, the use of ammonia bisulfate, and so forth. And finally, I would like to mention microbiological detoxification, which is actually, besides the use of adsorbents, which is also used by many companies which has also, however, the potential of absorbing also potential important or health beneficial components such as vitamins. So that has always to be carefully investigated. Microbiological detoxification has already become state of the art with bacterial cultures which have been subject to research for a couple of decades, yeast cultures, and nowadays actually also the use of pure enzymes to remove mycotoxins, especially from feeds, and also in collaboration actually here on site with Biomine, fermentation of microbes which are able to detoxify mycotoxins is a way to deal with this issue by adding microbial feed additives. So that is just to sum up a few strategies on how can we tackle the problem. The final way forward in order to control the mycotoxin issue is certainly analytics, because in the first instance you will have to find out what are the contamination levels which are there, and then only in the second instance you will actually have to deal with strategies on how to reduce this problem. And as far as analytics is concerned, there will be a lecture which I will give in the afternoon, and I'm actually looking forward to that afternoon and to the discussion afterwards.